Good morning. This is video 14. We'll be looking at transcription, modification of RNA before it leaves the nucleus, and translation today. We'll touch briefly on some of the epigenetics topics, but the next video is the one where we really spend time on epigenetics. Down here, you'll see the free response that I recommend you try once you're done with this video. If you look at the file that was loaded, I kind of rearranged these numbers just a little bit. So if you printed off the file with the free response on it, if you printed it off, say, weeks ago, it's fine. Just make sure that you kind of check the numbers because I've shuffled these around a little bit. I've got a new version of the file on our Canvas page, but if you already printed it off, don't worry about it. Just look and make sure you're doing the right ones. So back in 2017, on version B of the test, they have a version A that they just call the test, and then version B. 2017, version B, number 3, that'd be a 4-point question. 2016, number 4, that'd also be a 4-point question. And then 2019, number 1, that'd be a 10-point question. When you look at the 2019 question, it's like A, B, C, D, E, if I remember correctly. And the last few parts deal with stuff that won't be on the 2020 AP Bio test. So we'll be focusing on A, B, C. I'll probably still, whenever we have our Zoom meeting, walk you through D and E, but ABC is what pertains to reviewing for the AP Bio test this year. In future years, it'll all be fair game, I'm sure. All right, there's a picture over here. You can't see it very well. That's okay. What you can see is that the top one is a prokaryotic cell and the bottom one is a eukaryotic cell. So prokaryote or prokaryotic means before the nucleus kernel, so prokaryote. Prokaryotic. And then eukaryotic means true nucleus. And so one of the differences is with transcription and translation, if I don't have a nucleus, then as soon as I start transcribing DNA into RNA, let's say I'm only 5% done, once there's a piece of RNA out there, ribosomes can land on the RNA and start translating it into protein, even though I'm still transcribing. So it's not a 1-2, it's more like get started on transcription, then get started on translation. The analogy would be if I was gonna transcribe a 100-page book, I'm just gonna copy it, like freehand copy it, and then you're gonna read it. When I'm done with page 1, I can give it to you and you can go ahead and start reading, translating, so to speak. And then I'm working on page 2 while you're reading page 1. So in a prokaryotic cell, transcription starts and then translation is happening as we're still finishing up transcription. In a eukaryotic cell, like what I have up here on the board, again, there's a small version here that you probably can't see very well. Up here, the whole board is a cell. This is the nucleus. And in eukaryotic translation, or transcription and translation, I transcribe inside the nucleus. So one of the things that we learned about the nucleus earlier was that the nucleus protects DNA from being damaged by enzymes that might gobble up DNA. Well, another function of the nucleus is I can transcribe my entire RNA before I translate it. Then once it leaks out, now I'm in the translating room. So now I have a separate room for transcription and then later translation. Because of that, that means that I can actually edit the RNA before it leaves. So I might have a piece of RNA, but I could clip out certain parts, splicing we'll look at again, before I let it leave. And so that means one gene, one piece of DNA, depending on how I splice the pieces together, I might be able to make a whole lot of different proteins, alternative splicing. So that's a second function of the nucleus is now I have a transcription room I can edit, and then I can translate out in the cytoplasm. All right, so up here, the first thing you'll notice is my DNA is wrapped around histone proteins. Let me pull up a picture of that. So these are histone proteins. My DNA, you'll notice 1, 2, 1, 2. We get about 2 rounds of twisting around histones. And the idea is this. If this is my DNA, and it's all, I'll go from this end, it's less messed up. If this is all double helixed together, then whenever I want to read DNA, I have to be able to unzip it. So, let's see if I can hold this with my thigh. So when it's time to When it's time to read this DNA, then I need to be able to unzip it, read the gene, make the piece of RNA, and then ship it out for translation to build a protein. Well, if my DNA is strewn about kind of loosely, I can unzip DNA. But if it's wrapped around histones, then now that DNA is more in an off position. So wrapping around histones keeps my DNA from getting tangled up, but it also allows my DNA to be off. Right here, when it's wrapped around histones, I can't unzip this very easily. So when I want to turn a gene on, again, this is a topic we discuss further in the next video. When I want to turn it on, I loosen it from the chromosomes. I remodel the chromosomes, we say. Okay, so I've got histones there that my DNA is coiled around. Then I have two genes. Up here, these are protein coding genes. You can kind of get online and see about the argument of what's the definition of a gene. If a segment of DNA codes for a protein, we'll call that a gene. And that's how we used to define gene. Then they're like, well, we found some pieces of DNA that don't code for protein, but they still have a purpose, they have a use. So let's call those a gene. And so kind of a general rule now, a gray definition, so to speak, of a gene that I'll use is it's a piece of DNA that's useful. It might code for protein, it might be an on-off switch, something like that, but it's doing something. So I've got a gene up here that's coding for some protein. This is all in red. I've got another gene over here in purple. One thing that's interesting is that you would think all the genes that code for heart would be just in sequence, one after another. You know, chromosome 1 has more than 1,000 genes on it. So you would think, if gene number 1 is heart, then gene number 2 would be a heart gene, and 3 and 4 and 5 and 6, like that. They're spread out all over. And one of the reasons is gene number 1 might actually help you build a heart and build a brain or a lung. So they're kind of multifunctional. But the thing that's sort of interesting about eukaryotes is I might have a heart gene right next to a bone coding gene or something like that, or muscle next to bone, something like that. So that's a little bit surprising. It seems disorganized, but it's not. Okay, so in a gene, that's an R, that's a P, and that's a T. That R, hopefully it's not confusing, it doesn't stand for repressor, it stands for regulatory sequence. And so it's sort of an on-off switch. And the on-off switches will have names that sort of sound like they're going to code for on or off. So if that sequence is an on sequence, they call it an enhancer. If it's an off sequence, they call it a silencer. And enhancer sequences bind activator proteins. Both of those sound like I'd be turning the gene on. If it's a silencer sequence, then it binds repressor proteins. Both of those, silencer, repressor, sound like they would kind of turn the gene off. So that's a regulatory sequence. It can determine if the gene is on or off. This is a promoter. And remember, promoter, parking place for polymerase. So a promoter is a parking place for polymerase. In eukaryotes, that happens to be RNA polymerase 2. So RNA polymerase 2 wants to land on this promoter, and it'll start reading the gene. When it reads, we'll unzip the DNA, and then it'll start making the corresponding piece of RNA. So over here, if this was T, A, C, 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 G, or something like that, then it's going to find the corresponding piece of RNA. I'll make that purple. So the corresponding piece of RNA would be A, U, G, 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 C. Remember, in DNA, the base pairs are A with T, G with C. I should say A with T and then G with C. And then in RNA, it's A with U and G with C. So I'm cruising along here. My RNA polymerase will land, start cruising along, and start adding in the RNA nucleotides. It knows to stop when it gets to the termination sequence. And termination sequence or termination site will bind terminator proteins, and that sort of says this is the end of the gene. So the example that I gave earlier in the year in class was if I have something like this. You can't read that because it's gibberish, but you can still kind of conclude, hey, if this works at all like English, then I probably have 3 complete thoughts there. That looks like a sentence, capital punctuation, capital punctuation, capital punctuation. So it looks like sentence, sentence, sentence. So I probably have 3 sentences there. That's kind of the analogy of regulatory, promoter, terminator, regulatory, promoter, terminator. So when we see a promoter, that's the beginning of a gene. When we see a terminator, that's the end. So there are certain sequences that are common promoters. You don't have to memorize them or anything. You can Wikipedia. There are certain sequences that we find at the end of genes. So for example, near the promoter, a lot of times we find TA sequences repeated over and over to top box. Sometimes when we get to terminators, there's a string of C's, some crap in the middle, and a string of G's. So there are certain sequences that just signal start here and end here, just like these. Begin, end, begin, end, begin, end. And this is important because this is how we estimate how many genes are in the human genome. So when they first finished the Human Genome Project, you can look it up, but like they kind of had a draft sequence around like 2001, 2002, and then more like a finalized sequence later. So we'll just say early 2000s. You know, there were estimates as high as 50,000 genes, somewhere in there. And now we think that there's like a little bit more than 20,000 genes maybe. And one of the reasons that that number has kind of come down over the years is because at first it seems like I don't know what this means. I might not know what this gene is. I just know that looks like a promoter and that looks like a terminator. So question mark, I don't know what this is, but I think it's a gene. Same thing here. I don't know what these are, but I think it's 3 genes. Well, then you start getting into sequences that are like, you know, I went into Mr. Cotton's class yesterday. It's illegal, we're on quarantine, but whatever. Plus you can also see why I got a U unsatisfactory in handwriting when I was in elementary school. So here, I can read this and I'm like, oh, that's one complete thought. But if I was following these rules, I would see promoter, promoter, terminator, promoter, terminator. So it's kind of a mess, but I would think I have at least 2 complete thoughts. We go back, and once we understand more, we realize it's 1 complete thought. And so that's why sometimes you'll see the number of genes that we estimate in the human genome might change from year to year. Okay, so the next thing that we wanted to notice was when it's time to get all this started, how do I know that this gene is on and this gene is off? Because one thing that's interesting is I think I said a muscle cell and a bone cell. A muscle cell has all 23 pairs of chromosomes. So my muscle cell has, I'm just going to go ahead and say 20,000 genes. It's not exactly 20,000. So a muscle cell has all 20,000 genes, all 3.1-ish billion base pairs. Same thing with a bone cell. It has the same 20,000 genes, the same 3.1-ish billion base pairs. I'm going to now go with 20,000 and 3 billion. So why is it that bone cells grow bone and muscle cells grow muscle? Well, that's because the muscle genes are on in muscle cells and the bone genes are on in bone cells. But if I have a skin cell right here, it actually has the genes for growing muscle, bone, kidneys, brain. It just, those genes are off. Only the skin cell genes are on. Again, we'll get into that in a little bit more detail in the next video, Epigenetics. But one thing I did want to start with is when a gene is on, there are these things that will land near the promoter. Groups of proteins. And I'm going to put TF because they are transcription factors. And all these transcription factors like kind of land near the promoter, and they're basically saying, hey, this gene is on. So right here, I might have some transcription factors that become active and they go and they land at the beginning of genes. So I might start making a specific transcription factor, and it might not just turn on one gene, it might turn on gene A and B and C. When it's time to build a heart, if you're going to build this part, you also need to build this part. Kind of an analogy, I'll digress just a little bit, but if I'm building a car, if I want a windshield or just like a window for a door, like the glass window, then I need a door frame. And if I want a door frame, then I need whatever device is like putting that hinge on there, like those pieces of metal that like hinge the door. So there's really never a time that I would make a window without making a door frame, without also making the hinge parts. So if I make one of those, I'd make all three. That kind of makes sense. Now, there's really tons and tons of proteins that all like kind of glob on to the beginning of the gene. And some of them are far away, like here's a regulatory sequence, like this regulatory sequence right here. But I have all these various proteins that land at a promoter, and then a regulatory sequence might bind a protein. So if this regulatory sequence is called an enhancer and it binds an activator, it'll fold over, and basically all these proteins are globbing on near the promoter to basically say, hey, RNA polymerase, land here. Meanwhile, over here, we don't have transcription factors. We might have inhibitors binding nearby, and they don't allow the RNA polymerase to land. So the example I've given in class is if there's a parking lot with 1,000 spots in it, and maybe you're going to go visit someplace. We're out of quarantine, let's say. You're going to go visit someplace, and I want you to park in parking space number 312. Well, how can I force you to do that? I could just block all the other ones. So I could just go and put some sort of device, like a giant boulder, in every single spot. There are easier ways. I could just tell you park there. But in this analogy, I block all the other spots except 312, and you circle the parking lot until you find the only open spot. So if I'm RNA polymerase, if this guy is blocked, I can't land on there and read that gene. So I could land here. In addition, an activator is sort of like somebody guiding you. If you've ever been to a big event and everybody arrives at the same time or leaves at the same time, then you might have somebody guiding traffic. If you go to Six Flags, they're like, you park here, or a big concert or something, you park here, you park there. That's kind of what an activator is doing. So I block these so that they're off. These are open so they're on, but then I also have transcription factors and various proteins that are basically saying, land here, land here. That's what's happening near the promoter. So I'll transcribe my piece of RNA, and then it's essentially ready to be processed. So here's my piece of RNA that falls off. Now I'm going to draw it right here. I drew it in just purple, but I'm going to draw it in a few different colors here. There we go. Make that gap a little bit larger. And something that I think will show up on video. Green. So I've got purple, green, purple, green, purple. So now what we do is we want to process our DNA. If you look back at old AP Bio exams, these next few parts are important, especially the splicing. It seems like they ask that a lot. Whenever you get into these free response, you'll see it mentioned on 2 out of those 3, I believe. So what we want to do now is before this RNA can leave the nucleus, we have to process it or edit it. So this is what we call like an immature piece of mRNA. And we want to sort of finalize it and make mature RNA. And so one of the things that we'll do is we'll put on a cap on one end. If you can't read that, that's just 5 with a prime, like 5 prime. And so I put that on the 5 prime end. It's called a 5 prime cap. And a 5 prime cap, its job is it will allow the DNA to leave the nucleus. It adds some stability so that this RNA doesn't get chewed up. It kind of protects the ends. And it's also, once the RNA leaks out here and we do translation, it tells the ribosome like where the front is. So it's sort of like land here and start reading. So that's the function of the 5 prime cap. So that's one like RNA editing or RNA processing thing that we do. Another thing that we do, the third thing, is we put on a mini adenine tail, a poly-A tail, a polyadenine tail. We might put on a couple of 100 adenines, and that'll just look like this, a whole bunch of adenines. Again, it provides RNA stability once it gets outside the nucleus. It also allows RNA to exit the nucleus, and we'll also see in translation, it also acts as kind of a timer, almost like sand falling through an hourglass. If I've got my hourglass, as sand drops through, that's acting as a timer. If I put a couple of 100 adenines on here, it leaks out and we start translating it, the adenines start to fall off. So 200, 190, 180, 170, and so forth. Once it gets down to a small enough number, that RNA we're going to see will be degraded. And so it's like a timer for how long that RNA is out here. The longer it's out there, the more times we read it. Right? The longer some YouTube video is out there, the more views it can get. But once we take it down, it can't get any more views. So when the RNA leaks out, this will act as a timer. The second thing, and the reason I went out of order, is splicing is a little bit more complicated, but it's actually the second thing that happens. So my numbering system here is sequential based off of timing. So the next thing I need to do is splice. Splice and alternative splicing. So up here, purple, green, purple, green, purple. It turns out that some of these pieces are going to be spliced out. So what we'll do is we'll just name this particular purple piece right here R. Why? Just because. And this is A, and this is T. So the green parts we're going to splice out. And the way that we splice them out is essentially there are these complexes called spliceosomes. I've got pictures of spliceosomes in here. Let me pull up a spliceosome pic. So real quick, let me zoom in on this. So here's my RNA polymerase 2 in eukaryotes. It'll land near my promoter and it'll just start unwinding the DNA. It's going to cruise until we get to the termination site. Elongation, that's when we're just basically reading and making the piece of RNA. We're sucking in these nucleotides, A's, U's, G's, and CTP. So there's ATP, UTP, CTP, GTP. And the TP, just like ATP, whenever we're breaking off the phosphates and adding them in there, that's actually what provides the energy to build this strand of RNA. So we build this strand of RNA until we get to the termination sequence. Now I've got my piece of RNA. It's ready to be edited. So here we go. We've got a 5' cap. We're going to put on a poly A tail and we're going to splice. Now here, I'll show you a video in a second, the sequence. It's more like 5' cap, then splicing, then the poly A tail. So right here, they kind of already have the poly A tail on there, I think just for ease of understanding the diagram. But really, we do the splicing before the poly A tail. And so what we'll do is spliceosomes land on these green parts and they just splice it out. They loop them together. So the parts that are going to stay are called exons. The parts that leave are called introns. And students always find this annoying. I do too. I mean, we have exothermic. That means temperature. Thermic, heat, is leaving. Exo. We have endothermic. You know, we have exocytosis, endocytosis. Endocytosis is bring in. Endothermic is absorb heat. So you would think that like the introns would maybe like stay inside and the exons would be like cut out and they like kind of exit, but they don't. The exon stands for expressed and intron stands for intervening. Don't ask me why, they didn't ask me to name it. So exon, 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 I'm going to splice out those introns. So spliceosomes will land on here and they just sort of loop out the green parts. Like this. They call it a lariat, kind of like a lasso. I just say they're getting looped out. And essentially what's happening, you might remember my analogy that I gave in class. Let's say there's a boy over here and a girl over here and they're in love. And I'm the third wheel, I'm just some dude in the middle. I'm holding his hand, I'm holding her hand. And, you know, he's like, hi. She's like, hi. Wish this dude would leave. And so what I can do is I can basically join them together. I'm holding his hand, I'm holding her hand. And I just sort of like, as I join them together, when they join hands with each other, nobody's holding my hand anymore. So I hold my own hands. And just go over here by myself. I'm an intron. And I was spliced out. So that's what spliceosomes do. They basically will grab each side of this intron and just sort of loop out the intron. So I kind of loop these together and I make again what they call a lariat because it looks like kind of a lasso. And I put these together. And so what you'll notice is I didn't just pick these letters at random. Now I'll be left with one complete strand that actually has meaning. Again, mine I just assigned the letters R, A, and T. However, not only do I splice, I can also do alternative splicing. And that's where I put them in different orders. So don't forget to mention exons and introns if you're writing a free response. Remember, we only have free response on the 2020 AP Bio test this year. So exons are expressed, introns are intervening, and they are removed. But I can also splice things in different order. Imagine that I've got like 20 different people up here and we're all holding hands. I could start like, you grab this person's hand, you grab that one, or something like that. And we rearrange. What if that beginning intron and the end of that intron, what if they join hands? Then now I loop out all of that in the middle, and my final protein would be R and T. So now I can splice differently. I can produce the protein RAT, or I can produce the protein RT. But I can also, if I have like dozens of people up here, I can be like, you grab his hand, you grab her hand, she grabs her hand, he grabs her hand, move them around, and I can start rearranging the order. So I could basically, with one gene, I could produce this protein, RAT, or I could put it in a different order, ART, or TAR, rat, art, tar. I'm using the same gene and I get different transcripts, which will make eventually different proteins. And so this is one of the reasons why you might be kind of surprised if it only takes a little over 20,000 genes to make a human, as far as we know today. Why is there only 20,000 instructions to make a human? I would have thought many more than that. Well, one instruction might be able to make more than one protein. So it's a little bit like, in the English alphabet, we've got 26 letters. Does that mean we can only make 26 words? No, we can make infinite numbers of words, especially if the length of words isn't limited to a certain size. So we just recombine them in different orders to make different things. Okay, so once I do that, then now that transcript is ready to leave. And I wanted to show you a video. This is from a cell biology textbook. Let me give credit here real quick. So Molecular Cell Biology by Harvey Lodish, and he teaches at MIT, and if I remember right, a lot of these other co-authors are also from MIT. Anyway, they've got an animation that I got off of the web, and it's going to have some extra details that you don't need, but I think that overall you'll see that the overall picture is useful. So I'll even zoom in on this a bit. So you can see the promoter sequence, the enhancer sequence. There's the promoter and enhancer sequences. And so now my RNA polymerase 2 is going to land over here on the promoter. And it's going to take off. Again, we aren't worried about naming all of these different proteins or the fact that there's a phosphate group there. So I'm going to start making my piece of RNA. And as I'm making the piece of RNA, these little enzymes here are going to put on that 5' cap that we were talking about, 7-methyl-G. Some other proteins kind of grab. Notice they're splicing. So these are the spliceosome complexes. And they're going to splice out the introns. There might be multiple introns. Then we start adding a whole bunch of adenines to the end of this tail, 150, 200-ish. I think I read in the book that it's usually between 150 and 200. Now we bind that, and now my piece of RNA is ready to leave the nucleus. Okay, and so that's where we're going to pick up. So now my piece of RNA leaves, so it'll look something like this, 5' cap on the end, and a whole bunch of adenines on this end. And we had some stuff, and then eventually I get to A, U, G, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. And so what happens now is you might remember that the genetic code is basically a code of triplets. So this is the genetic code. You don't have to be able to read this. Let me go back. Just now realized I hadn't fixed the camera back. There we go. Let me zoom back out a little bit. There we go. Sorry about this, give you motion sickness. Okay, so my RNA leaks out with the 5' cap and the poly A tail, and my start codon is AUG. If I look at this, I know you might not be able to read it from where you're at, but AUG is the start codon for most genes. And so it turns out that A codes for nothing, AU codes for nothing, but AUG codes for methionine, the amino acid methionine. And so you might remember the idea was this. Let's say that I eat some, I don't know, almonds, and I basically take this, and let's say that almond protein are these 3 amino acids in this order. It's going to be more, but just pretend. So when I'm looking at my almond protein, it's blue, purple, red. That's almond. I eat it. And now what I do is I just rearrange it. Instead of being blue, purple, red, I make it blue, red, purple. And that's like muscle in my legs, and it was leg day or something like that. And so I'm going to build some protein. I just take the same building blocks from the peanut butter or the almonds that I ate, and I just rearrange them in a different order. So now I eat this, I digest it. These are all just in a random order. I need to somehow get this to land to where it's purple, red, blue. These 3 amino acids in this order. So what I'll do is whenever I eat these amino acids, I join them to a transfer molecule. Blue goes with blue, and red goes with red. So now, these are free floating around. So again, I still need to get it to land blue, red, purple, I think was my order. So if I can get these to land side by side, then these amino acids, 1, 2, 3 at the top, can join hands and build the protein that I want. So now I need to somehow get this guy to land in the front. Okay, so if he does that, there you go. One of the ways I could do that is if on the bottom of this transfer molecule, this is going to be a transfer RNA, if there was a UAC, then that would correspond with AUG. So my transfer RNA, I'm just going to make it look like this, tRNA, it's bringing an amino acid. Which one? The blue one. That's the one that I want. Down here we have UAC. So that guy's going to land in front. Well, if the red one was, let's see, CGG, and I want him to land second, then I need this corresponding code on the purple mRNA to be GCC. So essentially what's happening is if my DNA code is right, my RNA code will be right. If it's TACCG, my RNA will be AUGGCC. This will leak out, and now I'll have the correct RNA, which means the correct tRNA will land here, which means they'll bring in the correct amino acids, like this. So let me zoom back out a little bit. Right there. Okay, so let's say we keep on going, and then next was the purple one. And so if I want that to be GGG, then I need to have down here CCC. It's my tRNA. That'll bring in the purple amino acid. So again, the whole goal was I've got these amino acids from almonds that I ate. They're free floating in my body. I need to somehow get them to land blue, then red, then purple. Then if they could all bond with each other, then I've built my protein, blue, red, purple. How do I get them to land in the right order? I attach them to a tRNA, a transfer RNA. Those transfer RNAs are holding the amino acids up top, and they have an anticodon, a little triplet on the bottom. That anticodon, like UAC, matches the codon on mRNA, AUG. So now, as long as that AUG is correct, because my DNA was correct, then this guy will be first in line. So blue, then red, then purple. Then, as long as I can just get these guys to hold hands, I'm good to go. Now, eventually I get to a stop codon, like UA, UGA, and UAG. They're all kind of Tarzan sounds, are stop codons. And they code for a releasing factor. They don't code for a tRNA. That's how we know when to stop. So this right here is a codon because it codes for an amino acid. A doesn't code for an amino acid. AU doesn't, but AUG does because the code is always 3-letter words. Anticodon matches up. My tRNA brings in the right amino acid. Well, along the way, you might remember from organelles, I have ribosomes that help to hook up these codons and anticodons with each other. And so the ribosome is sort of like taking the mRNA, grabbing the tRNAs, and like sort of stapling them in there. I think of it as like a stapler. We sort of have this bottom small subunit and the large subunit. I'm kind of holding the mRNA like this, and then I'm just taking tRNAs and shoving them in place. So I do have a video clip of this happening, and I want to zoom back over to the screen so you can see that a bit. There we go. We're going to do this one. Skip forward a little bit. Okay. So I've got my 5' cap on here. That's my 3' end where I'll have a poly A tail. So there's the many adenines, my poly A tail. So we're now in this part, translation, happening out in the cytoplasm. These are my mRNA codons, my triplets like AUG, GCC. There's my methylated cap, my 5' cap. So the small ribosomal subunit's going to go until it hits AUG. Now we have a transfer RNA. There's a triplet on the bottom that's complementary to AUG. It's UAC. And at the top, it's carrying a specific amino acid. That amino acid happens to be methionine. Now the rest of the ribosome is going to land on there, the large subunit. This is going to show you that we have methionine. And technically, even though we start with methionine on a lot of these, sometimes the methionine gets edited out before we build the actual protein. The rest of the ribosome lands on there. And the job of the ribosome is, again, to hook up codons with anticodons. So the mRNA codons with the tRNA anticodons, as you can see right here. You might see that mentioned A site, a P site. You'll find books that also mention an E, an exit site. We join that amino acid over, the tRNA falls off. Slide over, join amino acids, tRNA falls off. This tRNA can get recharged. So as long as I either synthesize or eat the amino acid, I just recharge them. They're kind of like a mailman. They keep coming back. We just keep loading them up and they keep coming back. So I'm building my chain of amino acids, my primary structure of my protein. And then eventually I get to the end. So I'm going to get to a tRNA that codes for a releasing factor. Remember, my primary structure can fold up into like secondary structures. Okay, there's my stop codon, UAG. And now my protein is done. And then my protein can fold. Sometimes it will require a little bit of help folding. And it can go and hopefully do its function. If I go back to this, oops, maybe it was this one. There we go. If I go back to the Lotus video, 5' cap, put on the poly A tail. This is still inside the nucleus. Now we get to exit the nucleus. So you might remember that part that I mentioned, the adenines, the poly A tail acted as kind of a timer. And so various proteins are going to get rolling, and now we're going to do translation. So translation will be whenever the ribosome lands over here near the 5' cap and finds AUG, the start codon, and I just start translating. What I want you to watch are the adenines at the end. So over here at this end, you're going to see the adenines start to fall off. So adenines 160, 80, 70. Once they get small enough, once the number of adenines get small enough, then we degrade the mRNA. So again, that's like a timer. The longer that mRNA is out there, the more we read it. Many ribosomes can land on it. So as soon as one ribosome lands and starts moving, another ribosome can land and start moving along and translating also. From here, once we have this protein, then we want to secrete it from the cell. Based on timing, let's see, do I want to do mutations or secretion from the cell? So the quick version of secretion from the cell, let me zoom back out. The quick version of secretion from the cell, I'll kind of get into more detail of it in maybe a future video, would be like back to organelles. I take that protein and I shove it in the ER, the rough ER pinches off. Kinesin will grab that vesicle, take it to Golgi, start putting chemical tags on it, go to the edge of the cell, exocytosis. As far as mutations, what if I have a mutation? Is it a big deal or not? Depends on what the mutation is. So if I went back and looked at this chart, CCC codes for proline. CCA also codes for proline. I know you can't read this from where you're at, but CCU, CCC, CCA, CCG all code for proline. So if along in here, my codon, this genetic code or codon chart, CCC, that means this is proline. GCC is alanine. So if I changed a CCC to a CCA, that's fine, because I'm still coding for proline. So yes, there was a genetic mutation, but it's silent. It didn't hurt anything. Sometimes I might even change the actual amino acid. So some amino acids are big, some are small. Some are slightly positive, some are slightly negative. So let's say that I change the amino acid from a small, slightly negative amino acid to a large positive amino acid. That's more likely to have a major effect. If I change a small negative one to another pretty small negative one, that might not change the shape of my protein too much. Remember, when my protein folds up, part of the reason it folds up is because if that's a negative amino acid and that's a positive, then they will join. So as I'm building my protein out of my ribosomes here, positive, positive, negative, negative, oh, it might fold. And I start making like a beta-pleated sheet, for example, held together with hydrogen bonds. And so as I build my protein, I have it folding into the right shape. But if instead of positive, positive, negative, negative, if I have positive, negative, 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 then now I might like loop too early. And so that mutation might change the shape of my protein enough to where it has a different function. It's almost like keys. If I take a key and I, kind of file away a little bit of it, might not matter too much. Depends on where it's at. If it's on the handle, it doesn't matter at all. If it's in the part that goes in the lock, it might make a difference. With mutations, most of the names just make sense. If I've got a string of DNA nucleotides that's 100 nucleotides long, and later on I come back and I look at it and it's only got 98, so that means I lost 2 nucleotides, I deleted them, they call that a deletion. If I started with 100 and I look at it later and it's got 102, I inserted DNA, they call that an insertion. Sometimes in the middle, you just invert it. They call that an inversion. So most mutations make sense. About the only one that requires some explaining is a frame shift. And we'll end with this based off of time. Let's see. So this says, I think I read this in a textbook back in college. So the fat cat ate the rat. Again, remember in my genetic code, I'm always reading in 3s. So I basically go along and I say like, all right, the fat cat ate the rat. A frame shift mutation is like if I have a deletion and I delete bases that are not in factors of 3. So if I delete 1 base or 2 bases or 4 or 5 or 7, then now my reading frame is off. So let's say that I get a mutation, and let's say that I eliminate that C right there. So then I have the fat. I eliminate the C. I have a deletion. So now my reading frame is different. It starts off the same, the fat, but then it's atta, tet, her, and then that's not even a complete codon. So from the point that I got from the place that I got the mutation, a point mutation is a single spot. From the place that I got it, it messed up my reading frame. So I call this a frame shift mutation. Same thing would happen if I had eliminated a C and an A. If I eliminate a C, an A, and a T, then now, like I would still read it, the fat ate the red. At least it wouldn't screw up everybody afterwards. So a frame shift mutation is when, since we always read in 3s, when you eliminate a single or a double, something like that, then it changes your reading frame, and that changes the final protein. All right, the next video will focus on epigenetics, and we'll see if we can get all that into one video. Thank you.